No reason to stand up in front of it. So this is Dr. Davis. You know me, I'm Martha Noah, K-12 engagement coordinator. Dr. Davis has come from, is it the veterinary clinic?
No, I'm in the diagnostic medicine and pathobiology department.
There you go.
So it's within the College of Veterinary, we have 3 departments. One is clinical sciences, so a lot of those faculty have appointments and responsibilities in the hospital. Another one is anatomy and physiology. Those tend to be a lot of non-DVMs that do a lot of the early first and second year teaching, particularly first year teaching in physiology, anatomy, and then they do a lot of research and various questions in those areas. And then the last department is diagnostic medicine and pathobiology, which is everybody else, all the production medicine people, all of the path people, pathologies, which I'll be talking about today, and microbiologists, virologists, bacteriologists, all these different sorts of people. So some of us will teach in the DVM curriculum, but we also have a substantial graduate program as well for masters and PhD in vet med. And some of the people in that program have done their veterinary degree. Some of them go straight into that from an undergraduate because they're looking to do a degree in a very specific area, such as bacteriology of E. coli, for example. That might be what they choose to do. So I'm in that department. It's a real mix of people. It's an exciting place to be.
Well, it sounds like fun. Let's go ahead and get started then.
So I titled this because basically I'm this incredibly curious person who likes to do all these different things. And as you'll see, I've had other careers before I went into veterinary medicine. And so I'm going to talk about how I got to wanting to be an investigative veterinary pathologist. So we will back up what that is and how I got there, and then a wider view on the types of things we do in that field and how you can get there through multiple channels. It doesn't have to be the same way I did, through a veterinary degree and a PhD and a residency on top of that. So let's see if this works. So this is basically the agenda I went through. Who am I? What is this term? Investigative veterinary pathology. How did I get there? I'm going to go through, I thought best for this audience, or for you, to go through this from the point of view of the undergraduate students in the lab and what they're doing. So I employ undergraduate students in the lab for research. I also have a technician who would technically be a post-baccalaureate, meaning she has her bachelor's, but she doesn't have a PhD or a master's or any other higher degree. And we work as a group on both primary research that I lead and a lot of collaborative research with other people in the vet school and also at the National Institutes of Health, which is human medicine. So it's kind of an exciting place, and they all have their own projects. So I thought maybe the best way to introduce what's going on in my lab is through their projects and their lives, as opposed to everything I have to think about when I sit down at my desk, which can be even overwhelming for me. And then what next? If you're interested at all in investigative pathology, either doing it a little bit more within the veterinary medicine curriculum or as a career option, and I'll talk about some other options, talk about how to get there. What's the typical path, including some professional societies which do support even high school students and programs for high school students up through graduate student level. And they have a little advertisement about a program that's coming up next year in Chicago. So this is my official title. I'm an assistant professor, which means I'm on tenure track. So that means the university hires me and I go on a probationary period. It's not probation like I have a lock on my leg and they don't let me out of the house. This is probationary period in the sense that, and it's two-way. I haven't made a long-term commitment to the university, and the university hasn't made a long-term commitment to me yet. Each year we go through a reappointment process where, based on a plan of work I designed at the beginning of the year, I present to them, have I met my metrics, and if not, what have I done instead? And I go up for an approval process from the tenured faculty, people who have been there longer and who the university has decided to lock in a more permanent fashion. And you tend to be on tenure as an assistant professor for around 6 years. There are ways to cut it a little shorter, and there's ways to extend it, but that's the average. At K-State, and it's the average elsewhere. We have assistant professors who are called research assistant professors, or teaching assistant professors or instructors. Those are people who aren't on tenure track. They're hired specifically for a job on either hard money, money that's guaranteed by the state of Kansas, or soft money, i.e. they write a grant, and someone decides they're going to fund them for some research, and that's what guarantees their job. They're also up for annual reappointment, but it's a mechanism where it's done each year forever until they change their status. So that's, if you hear the concept of tenure, what's tenure? It does vary from university to university, but in general, it's as I just defined it. experimental pathology, which I'll explain, is different from diagnostic pathology. When most people think of a pathologist, they think of the person who received a cadaver, whether it's a dead dog and we don't know why, or a human that's died, we don't know why, I'm doing an autopsy, or a piece of tissue. You take your pet in and there's a lump, and the veterinarian takes it off, sends it in, pathologist, diagnostic pathologist. Experimental pathologist is involved more in exam research to solve a scientific problem. I'll get to that more. As we mentioned earlier, diagnostic medicine, pathobiology, that's the department. So for those of you joining me, you're all high school students, is that right?
Yes.
What year are you?
I'm a senior.
Senior?
I'm a sophomore.
Okay.
Freshman.
And you're all at same high school?
No, we go to Sherman West.
Okay. And
Olathe South.
okay, and we have Olathe in North. Okay, so I was just basically walked in, just introducing myself. The agenda I'm going to do is right here, just so you get a glance of it. And basically, we can take this where you want to go. This is a very small group, so feel free to interrupt me with questions and anything else you might have. I do have slides, but I can also go off the map quite easily if that's useful to you. How many of you are considering a career as a veterinarian? Okay, as a human doctor, as a scientist of some sort? Came here because this was the best thing to do with your afternoon. Not for fun.
I'm here because my sister's from here.
Excellent. Why are you here?
I like reading and figuring out just random things about.
Okay, so problem solving and things like that.
Well, I go web searching just for anything that I find interesting.
But you might find one of my prior careers interesting as well. So hang in there and maybe divert a little bit. So I was just introducing what my current title is, what I do on a daily basis. I'm faculty at Kansas Veterinary Medicine, School of Veterinary Medicine at Kansas State. And we have 3 departments there. One of them is diagnostic medicine and pathobiology. It's a really mixed bag of people who study bacteria, viruses, production animal health issues. In my case, pathology, which is the study of disease. And we're involved in teaching, extension, and research, as well as running the state lab and also helping the hospital. In my case, I'm involved in research and teaching. And this particular group here, CZAD, is Department of Homeland Security. So this is a federal government entity, a center, that is based at Kansas State, and it is chartered to look at emerging and zoonotic diseases in animals. So zoonotic disease is a disease that can move from animals to humans. And emerging disease is one, just as you might imagine, it's just appeared, or we sometimes have re-emergent diseases. For 50 years we didn't see this disease, then all of a sudden it comes out of the woodwork. Can anyone think of an emerging disease that's been in the news lately? It's a big virus, begins with a Z.
Zika?
Yes, okay, so Zika is considered an emerging disease. It's mainly a human disease. And there are some models for it in animals that are used to study it, but it's a massive human concern right now. Does anyone know why it's a big concern? Following the news at all?
It's very contagious, isn't it?
It's contagious. It's vectored by mosquitoes, right? So anywhere where there's the right species of mosquito that can carry it, it can be. Do you know where it is right now? Where they're having the issues? South America, right? So the mosquitoes in South America. Do you think there's a big fence between South America and here that prevents them coming in? No. So we share populations of mosquitoes with the Caribbean, where this is right now, and they come up onto our Gulf Coast and Florida. We have a number of diseases that over the centuries and actually decades have incurred into the Gulf area. The one that the Center of Disease Control was invented for in the 1950s was malaria. We eradicated malaria from the United States, but in recent years we've been dealing with a virus called chikungunya virus. Chikungunya is named after the horrible way you feel when you have it. It gives you crippling arthritis. Dengue virus, which goes to your brain. It's not anything you guys want. And the most latest one that we worry about, this mosquito vector, is Zika. And the main concern with Zika is that for a pregnant woman who's infected with Zika, it can lead to damage of the fetus, so brain damage. So that's in the news right now. What the Center for Emerging Zoonotic Animal Diseases does is looks at diseases that are currently outside the United States, or emerging in the United States in one place and possible spread. And what we can do, what vaccines do we have on hand? If we don't have vaccines, let's make new ones. What therapies do we have on hand? How can we control the spread of this disease, and how can we educate people so that they can recognize the disease and respond to it? So that's what that center does. And I function as the pathologist for that center. So I actually look at the tissues from animal studies and tissue studies. And then I do a little bit of teaching. This would be an example of some of the teaching I do, but I do other lectures as well. So that's my function at the university. And all of you, I'm sure, are very savvy on the internet, including, what's your name? Ryan, who likes to wander around the internet. So if you wandered around the internet and you typed in my name, you typed in KSU and maybe CVM or DMP if you knew the department, this is what you get. It's rather dry, right? This is my formal bio page. It lists a whole bunch of degrees up here. Mine are a little unusual because it starts with computer science and then middle school science, it's education, and it goes to a DVM and a PhD. It has some contact details, and then it has a bit in here. And what the take home of these paragraphs would be that, basically I'm a research scientist, and I look at diseases. I look at influenza, everyone knows what flu is, right? And I look at something called Rift Valley fever virus, which is a scary virus that can kill humans, but mainly affects cattle and sheep in Africa. And it's only spread as far as the Arabian Peninsula. So it's one of these viruses, these potentially emerging viruses in the US, could come across like West Nile virus did with mosquitoes, mosquito vector. And we don't have any good vaccines for it right now. So what we're trying to do right now is make vaccines for it so that in the event it does arrive, we're prepared as a country. So that's what I work on. I also work on a fungus. And the fungus I work on is probably inside everyone's lungs. Maybe one person would be free of it. It's what we call an opportunistic pathogen. Have you heard that term before in biology? Opportunistic? What does that sound like to you guys?
It becomes pathogenic when it has the chance.
Right, so in your nasal cavity right now, there's bacteria. In your lungs right now, there are probably pneumocystis fungus. They're little yeasts. They tend to hang out. They do nothing. You're normal. You don't feel like you have a pneumonia. You're walking around. Unfortunately, for patients that undergo transplants, for HIV positive people who get full-blown AIDS, for anybody who's under cancer therapies, those types of things, your whole body is immunocompromised. And then these opportunistic infections, whether it's bacteria on your skin and you cut yourself and it gets in, or whether it's in your nasal passages and the colony grows in there, or whether you have fungus that takes over in your lungs, that's opportunistic pathogen. So I also work with an investigator at the National Institutes of Health in DC, and I act as his pathologist. So I look at his tissues and cells through a microscope, and I help to determine for his therapy development whether the therapy is successful or not. And he got his start, this is Joe Kovacs, got his start as an AIDS critical care medicine doctor taking care of AIDS patients. And he saw that all these AIDS patients were dying from pneumonia, and they figured out that the cause of that was this fungus, and he followed that. So for the last 20 or 30 years, that's been his real focus. And so in joining him, I've been able to add some value where he can have a pathologist on his team. So the take home from this page is that I have a really kind of diverse job. And that's where I get back to my original title on this, License for Curiosity. So hopefully at some point in the next few years of your career, as you're traveling through high school and going on to college, you're going to find a subject matter that catches your attention. You may find multiple subject matters that catch your attention, but a question that goes across them. So an interdisciplinary question. Maybe it uses math and biology. Maybe it uses languages and looks at an ethnic tribe somewhere in Central America. But you're going to find a question you pursue. And then you're going to want to find academics that support you in becoming good at that area, whether it's medicine or whether it's fine arts. And then you're going to pursue that. And some people pursue things in a very deep manner, and some people pursue things in a very broad manner. And even though it looks like I've kind of gone on this veterinary theme and stuff with a PhD, and it all kind of is about infectious diseases and pathology, I would actually still be viewed as very broad. So whereas somebody in microbiology might focus on a single protein in a bacteria, I focus on several different viruses and a fungus. And what I tend to do is look at the interaction they have with tissues, and where are the connections with how they do it similarly and different across those diseases. So it's kind of a, not a compromise, but a way to kind of stay broad, but and also have the opportunity to go deep for periods of time and then come back again. And for me, that's where I love to be. I love to have things I don't understand, and I have to do lots of reading and figure them out and figure out how to solve the problem, and things that I just know how to do that. I bring that to the table, and we can take how this problem's been solved in this space, walk it over here, and apply it again. So we did that with flu. Have you thought of trying that with this other agent, this fungus that causes pneumonia? Flu causes pneumonia, so does the fungus. How can we look at things in the same way? So that's basically what we do. So if you actually start clicking on these links, there's one down the bottom here that says PubMed search. That's code for what have I published, right? And that lands you somewhere like this. I don't know if you guys have ever delved into the internet this far, but this looks like I've been very busy writing papers that have very long names, where you probably have to look up quite a few of the words. So basically I'm published, that's what it tells you. If you go and read these are primary research articles. And so they assume when you go into them that you have some basis in biology and in virology, and in some cases pathology when you read them. But at least from the titles, you can see that I've worked with a diversity of animals. Here's a sheep, here's a human, here's a non-human primate, some of these are mice, some of these are birds. So I don't seem to stick with one animal, and I don't also stick with one pathogen, one virus or fungus, etc. One of the things I've worked on that often faculty at university haven't worked on is a LinkedIn page. And so this is more of a professional presence. And one of the things I try to do here is showcase work that my students have been doing. So in this case, this is we'll get to Lizzie later, but she just won an award recently for some work she was doing in my lab. She's a senior who's progressing on to vet school next year. So this is a little more, how shall I say, friendly. So if you want to get to know faculty in universities, don't just go to their department page and stop. See if they have a presence on LinkedIn or on any of these other professional networks, Plaxo, maybe Facebook. Some people have got to Facebook. They're not usually out there, and there aren't too many faculty tweeting, but perhaps, all right? So, but this stuff's out here, and more it's more accessible in terms of what's going on. So my title was Experimental Veterinary Pathology. That was selected by the university. That went on a job ad, and I responded to a job ad, and I got that title. I actually prefer Investigative Veterinary Pathology. And so what is this? This is a branch of medicine. If we take the veterinary piece out, we're talking about the nature of disease. So when a pathogen is interacting with an animal or even a plant, you can have investigative veterinary pathology of plants too. We're looking at what changes does it make in the tissues and in the cells inside those tissues. And then more importantly, we're looking at how is it doing that? What's the mechanism, the cellular biology mechanism causing that, or the interaction between the pathogen and the tissue? So some pathogens go inside cells and some of them stay out. Viruses have to go inside cells. Hopefully if you've had any biology, you kind of understand that viruses are dependent on cells. There's a whole debate, and this could be a debate that could go on for hours, as to whether viruses are alive or not, right? They're only really alive when they're in a cell replicating. If a virus is lying on the carpet here and there's no bacteria, it's inside, it's effectively dead. It can't do anything to you, it's inert. But the moment it comes into contact with a cell that has a receptor that it can bind to help it get into the cell, then it's alive, right? It can replicate, it can do things, it can multiply and leave the cell. And when it leaves the cell, sometimes it destroys the cell and sometimes it doesn't. Whereas the other pathogen I mentioned earlier, the pneumocystis that causes the fungus in the lungs, that's extracellular. So as far as we know, it can't get in the cell. Although one of the interesting things we're looking at is perhaps it connects itself to the cell. And how does it do that? So that's one of the areas of interest. So not only are we interested in what happens to the tissue, but how that happens to the tissue. That's the part that I'm particularly interested in. And you add veterinary to this, which means you've added an animal medicine side of this. So I work a lot with investigative pathologists who are in human medicine, and they use animals as models, but they don't have the broad training as an MD about all the different animal physiology, anatomy, and animal systems that you would get if you studied that specifically. And they're MD veterinary, they're MD pathologists as opposed to. So these are some of my scopes. This is actually my office over here. This is a bright field scope. It's basically an enlarged version of something you've probably used in your classroom, right? It's got more lenses, it's got more power, more light, and it's connected to a camera. But in the end, it allows me to look at things I can see with my eye, visible light. Whereas these scopes over here are fluorescent. Have any of you ever worked with fluorescents or seen fluorescent scopes? Okay, so basically, from a fluorescent scope, the background of the image is going to be black, and then there's going to be some bright colors on it. If you remember back to the first slide which you saw, Allie, it had this one here. That's a fluorescence image. In fact, this is a piece of human trachea. This is the lumen where the air is going down. And then this is the epithelial surface. And in your trachea, you have mucus cells. This cough, right? What comes up? Mucus. So your cells that make that, they're called goblet cells. And you also have ciliated cells. Remember the mucociliary escalator, which is when something goes down that shouldn't go down, it's what helps push it back up again. You cough and it comes out. And these are the different types of cells. In green, I've labeled the mucus cells. I've used a special cell marker to do that, to make them green, and lit up the trachea accordingly. There's actually some pink here too, which you can see better on this side. This is actually a trachea that's been infected with flu. So I've used a marker to find the flu in that one. So that's a fluorescence image. So you use a different type of scope to get it back. There's some other fluorescent images back there too. So I use experimental pathology techniques, which means I make a lot of slides of tissues and cells, and I look at that on scopes, and I add markers to those slides to help me light up certain things of interest, whether it's a cell type, whether it's an organism, whether it's a change. Maybe an organism leaves iron behind. You know, some of the bacteria like to do that. Maybe it destroys something in the cell, and the cell leaves something behind, like a product. So you can find all of these things. Basically, the footprints of the interaction between the pathogen and the host. And I talk about comparative pathology here because my training allows me to think about, well, this happens in a human. In a mouse, we also have that structure. We also have a lung. It's a little different, but we have it. And so what happens in a mouse lung by comparison? So comparative pathology. And I didn't take pictures of it, but I also have what you would view as a more traditional lab bench where I can run chemical reactions and create slides and all these types of things. And so that's what I work with on a daily basis. And this is not responding. Now I wanted to give you guys a chance to show me if you can see something. So what's critical to my job is being able to, actually I'll go back. What's critical to my job is being able to see things. So take a look at this picture here. First of all, can anyone guess what organ this is?
Liver.
It is the liver, yeah. Okay, so which one do you think is normal? The one on the left or the one on the right? This is sheep liver. It's a little bit different shape than a human liver. Left, yeah. So it has what we call the classic mahogany color of the liver, and it's a bilobe structure in the sheep here. The one on the right, a couple things you can do. How would you describe it? What's different about it in terms of color?
It's really pale.
Pale. Anything else about it? Anyone else see anything on that one?
It has like a film on it.
Yeah, it's hard. That's why I tried to look at it. So you see the little dots, little tan dots on it? Yes. So it's a little bit like a film. It's actually not a film. There's a lot of glare on this. This is taken within a facility that doesn't have great lighting, unfortunately. But what that is each of those little foci, those little tan foci, that's basically an area where the tissue has died. So this animal on the right was infected with Rift Valley fever virus, which the first organ that it attacks is the liver. And it actually causes cell death in the liver, actually whole tissue death. And you can see them. They're 1 to 2 millimeter foci that appear not only on the surface, but if you cut this liver in half, they're throughout. It's almost like the liver has been polka dotted, right? Tan polka dots throughout the entire thing. And absolutely right, as Ali said, very pale, right? How about this one? What's different about this one? I've kept the same one on the left, but the one on the right is changed up. And this one's inside the animal still. So this is the stomach here, actually. So just ignore this here. That's the stomach, and that's part of the mesentery and a bit of the intestine there. So the liver is this piece here. And it's underneath the diaphragm, which is where it sits in a human too. So compare it to the last one we had here. This one we said was pale and had lots of tan foci, little polka dots. See anything new in this one?
It definitely looks like it's infected with something. It definitely still has like spots on it, but it looks different. Like either maybe it's not as diseased.
So what color are the spots this time? You see them?
They're more purplish, like dark.
Yeah, they're actually red. Yeah, in this case, it's their blood. This is actually, you did notice differences. This is actually, I tried to put a star on here to help us out. See the star right here? See the large tan foci? So instead of this being less disease, this is actually more, because what's happened is the tan foci are larger, and where they got a little larger, not quite this big, but sort of an intermediary size, so much cell death occurred that then where you have space, blood flows, right? In your body, particularly in an organ like the liver, because there are loads and loads of capillaries all over the place. We call them sinusoids in the liver, where the blood's flowing throughout. If you take away, then it fills with blood. It's just like if you poke yourself, what shows up, right? First thing that runs into that is blood. So if you compromise any vascular structure, poke yourself in the skin, cut yourself on the knee, blood goes there, right? So same thing in the liver. If you have damage that's killed off some of the cells, then blood flows in. So this is actually further along. This is a more diseased sheep liver in that case. Okay, so pretty down the gross, and this is what gross pathology is all about. It's just about looking at the images and making observations. And the more you do this, the more you understand that if you see tan foci, that's either something added or something lost. And then you can look at the overall liver itself, and if something's added, things look bigger or bumpy. And if something's lost, things tend to be depressed a bit. It's a little hard to appreciate some of that from a photo, but these are actually not only tan foci, but they're a little bit depressed and dull looking, so something's been lost in this case. Something added would be, let's say there was lots of bacteria there, little foci of bacteria, or let's say there were lots of extra cells that came in from the immune system to respond to a disease. In this particular disease, which is a viral disease, this animal's only been exposed for 2-3 days. There isn't a lot of time for that. What's happened is just massive cell death. And then where do we go from here? So once you have this sort of gross impression, you need to actually look at it under the microscope to confirm what you're looking at. So let's look at this one. So the one on the left, neither of these is normal, but the one on the left is more normal. This is the central vein. So your liver, how many people have looked at liver before and different parts of liver tissue? Any of you? Sir, Henry? So basically, think of a bunch of hexagons. You know, like a bathroom floor? Got a bunch of hexagons all on the floor. And in the center of the hexagon is what we call the central vein. It's where the main blood flow leaves the liver. So the liver is basically a stack of these hexagons in all dimensions, 3 dimensions, forming that lobular structure you saw earlier. And we've zoomed into one of those hexagon centers. And in the center of that, there's a vein. It's perfectly normal, has blood, drains out of the liver. Surrounding that are this structure here. This is a bile duct, which goes down to the gallbladder, which we saw in the previous picture here. And a gallbladder is involved with making bile, and bile goes into your digestive system. And so when you eat that Dairy Queen Buster Bar or that really, really fatty burger from McDonald's, hopefully you don't do this too often, but basically you need something that breaks down all the fats. And that's what bile does. So that's released into your intestine. So bile is first made in the liver. Many of you in anatomy or physiology, so it'll help you out. So bile is first made in the liver, and it starts out in these little ducts, and then it flows down to the gallbladder. And most animals have a gallbladder. Trivia question, does anyone know an animal that doesn't have a gallbladder? There's usually 3 that are really easy to remember. Horses don't have a gallbladder. So basically they make bile in their liver like this, and then it flows directly into the intestine, kind of on a pulsing type motion. Rats as well, no gallbladder. And the other one I like is dolphins. And I remember that one really well because I was tortured by a mentor one time. I was doing a dolphin necropsy. I used to work on stranding networks both in the Northeast and the Southeast of the United States. And one of my mentors challenged me to do this dolphin necropsy, and he challenged me to find the gallbladder. I spent a long time rooting around this dolphin, trying to find the non-existent gallbladders. To this day, I will never forget that. But all mammals are able to make bile. They just don't all have gallbladders. So it's a good party question for you. So they all have these structures called bile ducts, which are part of the production system for making bile. And the other thing they have in here are arteries veins and arteries, as well as lymphatics. Everyone know what lymphatics are? Lymph nodes, right? So you have this drainage of lymph, which is another humor. If you remember back to ancient medicine, we had different humors. We had urine and blood and lymph. These are all things they like to drain out of people in the Middle Ages when they were sick, because they thought, you clear all the humors out of the body and you'll get better. Just use it. They're back to using leeches in medicine, but the idea in most cases when you had a disease in the Middle Ages was, bad humors, have to get these out. So they drained every fluid they could think of out of the body. Lymph is one of those. So in all the organs of the body, we have lymphatic flow, and that's true in the liver as well. So back to where I was going with this. This is normal in the sense that this is a normal hepatocyte. Not only is the liver made out of big hexagons, it's also made out of little hexagons. So we call these cells polygonal cells. These are the normal hepatocytes. And they're happy and fluffy red with a nice fluffy blue kind of looking nucleus at the center. So draw a hexagon, put a little nucleus at the center. Normal. And then those of you who are astute will have noticed there's all these little blue cells all over the place. And these are cells from another system in the body called the immune system. So they're lymphocytes. So if I drew blood on anybody in here, you'd have red blood cells and you'd have white blood cells. And lymphocytes are some of your white blood cells. And they're responsible for responding to allergens, to pathogens, to any sort of disruption in tissues of your body. And so in this case, these guys are responding to something. And that what I would call, as a pathologist, I would call these low numbers of lymphocytes in this case, in this portal tract area. And then moving on, we have this one over here. What's different about this one versus that one? Still pink and blue, right? So that's first observation. Same pink and blue. More blue or less blue? More blue. So for pathologists, number one rule of pathologists, blue is bad. Okay, so if you remember anything from this lecture, I tell you the pathologist said blue is bad. Blue is bad because in cancers, that's a proliferation of cells, you get more blue. In infection, where lots of cells respond from your immune system, you get more blue. So basically, if I put a tissue slide under the scope, I know as a pathologist the average color I would expect in the liver, which is mainly pink. And if I put it under the scope and it's blue, then I know something's wrong. And then I start looking, to see, okay, what is blue? In this case, what is blue? So we have these, I told you these were lymphocytes over here. We have a few similar looking cells here. They've actually, it's hard to appreciate from here, but they've got a little bit more of a bumpy nucleus. They don't have a nice, perfectly round nucleus. They're neutrophils. They're another leukocyte, another white blood cell. They're all these white blood cells. I told you a hepatocyte was this nice hexagonal or polygonal shape with a nice fluffy blue nucleus. Do you see any of those on the other side? There's one up here. Okay. That's the only piece of normal tissue here. Oh, maybe these two down here. So basically this is really wrong. So if we go back to that image where we had the little tan foci, remember this? This is a close-up of one of those little tan foci. And this is basically a hole forming in the liver where all the cells have died. And if I came back to this hole, if this animal lived and I came back to this hole in another couple days, it might be filled with blood or it might be collapsed in. But basically these cells are gone and they're not coming back anytime soon. What'll have to happen is that if this liver recovers, these cells on the edges will have to divide and fill in this hole here. And each of those little tan foci that you saw in the earlier picture of the liver itself were related to that. So arteries, veins, lymphatics, and bile ducts, those are components in your liver. Proper hepatocytes over here, and then dead ones over here. And basically what a pathologist trains to do is to be able to look at this sort of thing in every organ. So it's all about pattern recognition and being able to identify what the causative agents of these different things are. And we use a lot of tools. And one of the tools we use is immunohistochemistry. And immunohistochemistry is a fancy term for being able to mark something in the tissue with an antibody. So an antibody is something that finds a protein in the tissue and latches onto it. And then we use interesting techniques in biochemistry to cause a chemical reaction and make a color. And so the color you're seeing in this tissue right now, basically the footprints of the virus. So these are bits of viral protein from the Rift Valley virus sitting in the middle of that lesion. So I go from lesion to lesion caused by virus with confirmation of presence of virus in that sort of change to this immunohistochemistry. So I do a lot of that for a lot of different types of viruses and pathogens, and I ask different questions and use different tools to look at that. I don't do it within the diagnostic pathology field. In other words, people aren't mailing me samples or sending me animals for autopsy or necropsy, if you use the term that veterinary pathologists use sometimes. I'm more involved with a virologist who's asking a question. So for example, the previous slide here was used most recently from, this is actually from a vaccine study, where we had some animals who received the vaccine and some didn't, who didn't. They were all challenged with the virus. And then it was very important to know at the end of the study whether the vaccine worked. And in this case, this is a control animal, so the vaccine wasn't on board with that animal and it has the normal full-blown disease. And this animal over here is vaccinated animal and it didn't have any lesions that were attributable to the virus. It had this other lesion here where it had a little bit of inflammation. And so I pursued that inflammation like a diagnostic pathologist at that point. And I went, how did that get there? And I went down in this animal's intestine and found a protozoa called coccidia. Coccidia, each different animal species has several that infect it, but it's a mild disease. But because it's in the intestine and the intestine's connected through the gallbladder and other systems to the liver, through blood supply, it can cause inflammation in the liver when the actual infection's in the intestine. So that's where that came from. So I just wanted to give you a brief background on me, and one of the things I wanted to get across with this is I did not have a direct route to veterinary medicine. And if that's where you're headed at some point in the future, you don't have to have a direct route there, or you can have a direct route. There's no harm or foul in either one, or benefit. I think what I did as a career is very useful because I look at things a little bit more interdisciplinarily than some of my colleagues. So I was born in, I'm Canadian by birth, New Zealand by family, and I started out in Canada and then I immigrated to the United States. I'm a naturalized citizen in the United States. And then in my early 20s, I worked in England. Does anyone know the flag in the middle there? Nope, that's the Netherlands. So I lived in Amsterdam. And the next one? Italy. Yep, so I lived in Italy. And this was all while I was doing a very different career, which I'll get to in a moment. So pre-vet life, very much like you, well, maybe the scopes look a bit better now, but I played around with microscopes in middle school and high school. I was interested in what I was looking at, particularly interested. I love drawing too, so I used to like drawing cells and everything through the microscope. I went to university, I went to Dartmouth, and a couple things happened to me in my first year or so. I got off course and I discovered I had a knack for computers, so I went to computer science, and I have an undergraduate degree in computer science. And I went and used that degree. This is a consulting company that's not around anymore, but needless to say, in the 90s when I was working in industry, they were very strong. I joined them when they were about 200 people. They grew to 4,500. It's a very large company by the time I left it. And we worked with all these clients. Some of these logos should be familiar to you guys. Many of these companies have changed logos. Kmart's still around, right? Yeah. And Swissair used to be a major airline in Europe, but it's gone under. This is a bit like a Sam's in France, Carrefour. It's what we call a hypermarket. And then there's a bunch of banks. This is an insurance company. And Hughes Aircraft, Lockheed Martin. This is aerospace. Point being that I worked with lots of different industries. So I got a chance to see what different businesses were like. I got out there, tried a bunch of different jobs on, basically, as a consultant, helping them with their computer systems. While I was there, I also learned to think in a systems way, about solving problems. And I find that that's very useful to what I do today. I was actually working at one of these banks. This is the headquarters of Britannia Building Society in England. And every day at lunchtime, a bunch of cows would walk around the building because the farmer had ceded this land to Britannia Building Society to build their headquarters, but reserved grazing rights. Very common thing in Britain. So you were working here, I was working on mortgages, and there's cattle here walking around the building. I'd see them go by. And then this was in the 90s, and so mid-90s. And so one day the cattle didn't go by, and the next day they didn't go by either. And I finally inquired. I said, where did all these cattle go? And they were actually on a funeral pyre. So in other words, this was during the prion disease outbreak in the UK. So it was called bovine spongiform encephalopathy, which is a prion disease. They're aberrant proteins. They're actually pathogenic. There are a number of different diseases, including some that are genetic in humans. Creutzfeldt-Jakob's disease has a genetic familial form. Familial insomnia is another one. But the linkage and the concern here went drastic when there was a link made between bovine spongiform encephalopathy, scrapie, which is a similar disease in sheep, and then moving forward from the cows to humans, something called new variant Creutzfeldt-Jakob's disease. And they linked the same prion protein, in other words, the same infectious agent across all of them. It evolved between each, but it moved. And so they actually yanked all the beef off the shelves in England. They banned exports of British beef, and this went on for a couple of years. And this is actually a news clipping from that time. Major was at that time the Prime Minister. John Major was the Prime Minister of England at that time. And I won't translate it for you. You can figure out what's going on, but it's a pun on basically they're ridiculing him in the newspaper, and the bulls refers to the cattle at the time that he had to go. And basically he was very upset with the EU or the wider Europe at that time because they were trying to get the trade barriers dropped again. There were huge trade barriers against England, and it destroyed their agriculture at that time for beef. And then this is actually where I lived at the time. So looking at this picture down here, it looks like a nice party atmosphere, and you wouldn't realize that all of this was going on at the same time. But the reason I put this up here is it gave me pause. There I was as a computer consultant doing mortgage applications and actually quite bored. And all of this was happening around me. And I thought, well, maybe I could use my brain to go back and solve some interesting infectious disease problems. So this was the turning point for me. So I actually left the career I was in. I took actually a unpaid position and a minimum wage paying position as a veterinary assistant just to see what it was all about. I had last been in a veterinary clinic from a behind the scenes point of view at age 13 when I assisted with surgery and observed. And so I was back in that environment, and I really enjoyed it. And this individual at the time who I worked with was also doing canine Lyme disease research. Lyme disease is also a disease that affects humans. I also took a position, unpaid initially and then paid, at the Mystic Aquarium. And through the University of Connecticut, I did research at the Mystic Aquarium on beluga whale blood. So I looked at blood values in beluga whales, how to measure blood values that had to do with the clotting cascade, so how blood clots in a beluga whale, as well as some standard CBCs and chems, so blood counting cells, and also chemistries that look at changes in other tissues. And I did that as part of a research course, basically an undergraduate level at the University of Connecticut Mystic Aquarium. These were prereqs. I decided at this point somewhere in here that I was very keen on veterinary medicine, if not veterinary medicine, then biochemistry. So I worked in the biochemistry lab as a research assistant for a bit. But, I was in my late 20s at this point, and I wasn't sure I wanted to go do a whole doctorate. So I went and did a summer internship for a month or so at the Brookfield Zoo with a nutritionist. And to be a nutritionist, basically, it's a master's degree, right? So I thought, I really like zoos, I really like nutrition, maybe I'll be a zoo nutritionist. I forgot to mention that initially I went to the Mystic Aquarium because I thought I wanted to be a SEAL trainer, but that did not work out. So here I am down here at Brookfield Zoo. So explore, right? So I'm over here, I do this internship. I'd been accepted to vet school at that point, or shortly after accepting the position at Brookfield. But I still went, and it was a pretty amazing internship, and it actually led to a presentation at a national conference on Western grey kangaroos. And so I looked at their diet in the wild based on what had been published in research, and then I modified their diet in the zoo to be closer nutritional value-wise to what they had. So that's what I did. And then I went to NC State, not K-State, but they're both veterinary colleges, and they're both very good. And while I was there, I focused on some of the exotics. This is a snake. I'm doing cloacal samples from a snake here. This is a sloth. I was responsible for monitoring the animal. It was under anesthesia. You don't want to do a physical exam on the sloth unless it's under anesthesia, because it's got an iron grip that can almost break your arm. So that's not worth doing. I went down to Honduras, and that's a coatimundi, which is a natural It's a relative of the raccoon, and it's native to Central America. And this is pretty standard. If you're going to go to vet school, you're going to have to stick your whole arm up the backside of a cat. So that's what I did there. And while I was in vet school, I did a summer out at Stanford, where this is where I first did virology research, and this is where I discovered my love of viruses. I didn't really want to be a virologist, but I wanted to somehow do a field that connected to that. So here I'm studying murine cytomegalovirus. It's a type of herpes virus, not the herpes simplex virus, but a related virus that actually causes mononucleosis. Do you folks know what that is? Yeah. So that's usually caused by Epstein-Barr, which is another herpes virus, but sometimes caused by cytomegalovirus. And we study it with the mouse version of the virus, which is closely related in mice. And so I was doing that here. And in fact, I spoke with this person on my way over in the car. I still stay in touch with her. This is the first investigative pathologist I met. So Donna's a DVM who did a PhD and a residency in path, but then instead of becoming faculty at vet school or working in a diagnostic clinic, she joined the faculty at Stanford, and she works with medical doctors on human disease models. And then there was an escape of frogs into a local lake in Golden Gate Park. It's in San Francisco. You guys know where the Golden Gate Bridge, there's a park near that. And there was this mysterious report in the local newspaper that there were frogs there that didn't look native, and it turned out they were xenopus frogs, which are used in labs. And so with this professor, I went and caught them, and we did disease and genetics work on them to see if we could figure out whose frogs they were and whether they were dangerous, because the local Chinese population who lived there, ethnic Chinese population, were catching them and cooking them for dinner. So we were a little worried that they might actually all get very sick. So that was a little bit of diagnostic pathology there. I'm going to skip past this unless someone's particularly interested in marine biology. We can talk about it later. But I took those ClinPath interests in blood and other fluids and did a whole piece of work in Miami with this organism here, which is called a sea slug. And we got cover on a journal for that. And then the other thing I did is, as I was in my DVM, I went to my DVM, I knew I wanted to do infectious diseases. I lived through this prion outbreak in England. I wanted to figure out what to do, but I didn't know what that meant. So during my DVM, I explored a lot of things. Public health, I explored pathology, and these are some of the things I did to explore pathology. You'll find in all walks, even if you choose any science, that there's always professional societies that support trainees. And so I went to PATH conferences with the American College of Veterinary Pathologists, and I also went to conferences with the zoo veterinarians, and I explored which way I wanted to go, clinical zoo versus pathology versus infectious disease. And I went to a lot of different institutions. I basically got invited to each of these places for short periods of time, a couple weeks rotations, mainly in my fourth year, but some of them before. And some of these are hopefully recognizable. Johns Hopkins, this is the National Aquarium, part of the Smithsonian. It's based in Baltimore, and a number of universities. And then I also picked up a USDA scholarship to go to Indonesia to do an epidemiology workshop. This was right at the end of my DVM before I started my path residency. And this is actually what sold me on this pathogen. And this is the guy I ended up doing my whole PhD on. So, but if I'd never gone to Indonesia, I might not have ended up doing that. So you have to sort of go where life takes you, is the message there. I did a traditional path residency. That's mainly training you to recognize lesions in tissues as a diagnostic pathologist. This one down here, I call my beach ball fish brains. These are actually a pathogen in the brain. This is a fish that came from a fish farm, and obviously we don't want to eat these fish if they've got these things inside their brain. And as I said earlier, stranding networks. This is a pilot whale. 32 of these washed up on the coast of North Carolina during my residency, so we responded to that. So a real mix of wildlife zoo as well as traditional diagnostic path. This is actually an intestine, but this little bumpy things here, not normal. They're diverticula, the little sacs that grew that shouldn't have grown on that particular animal. I went here, I emphasize this only because even at the high school level, the National Institutes of Health has training programs for research. For anyone who's interested in research, they have a good website for this. I hosted a high school student while I was there. They have undergraduate opportunities. They're competitive, but if that's really what you want to do and you're articulate about it, you can get there. And there's 27 institutes here, all focused on different areas of medicine. And this is building 10, which is the largest building on the campus. And it's where the pneumocystis researcher I work with is based. And I spent my years there doing my PhD as part of a special program for veterinary pathologists doing PhDs. And there's a couple people a year in that program. And this is the lab that is the base of it. This is the lab I did my PhD in. And this is a building which is designed for high containment research. So I don't have a picture of the BRI here because you have to get an official picture of any of these buildings to show them. But basically they're designed with massive air control systems to protect the people inside and out, and then also massive barrier systems in terms of physical security as well as dealing with access to the agents. Because in many cases, these are foreign animal diseases that we're developing vaccines for that we don't want any sort of release. I just wanted to spend the last couple minutes talking about the folks in my lab and what they're up to. This is not by design. I have a lab that's entirely women. That was not by design. We do competitive entry into the lab based on resumes and cover letters and even interviews. It just so happens that right now we're all female. So that's me in the center. This is my technician. She's been with me for a year, and now with my blessing, she's going on to do her PhD in molecular virology. So she'll be switching to another lab at K-State. And each of these people is undergrads. Maya is a freshman, Katie's a sophomore, and Lizzie's a senior. This is what Lizzie's been up to. She works with, this is Rift, a cartoon of the Rift virus, as well as the influenza virus. She doesn't work with the live infectious agents. She works with inactivated materials, and she helps me find things in tissues. And so she recently took her work with me to San Diego, fully paid trip to San Diego from one of my professional societies and from Kansas. She picked up $2,000 and came with me there and presented this poster. So this poster talks about the biochemistry of the stuff she's doing at the bench to make the slides, as well as the end pictures. And some of this should be familiar because I showed this earlier. But you notice here we use different colors to highlight different things in tissues, and we also do fluorescent work. So she's been in my lab for about a year and a half. She works on 2 projects, and this is the formal output of what she's done. And so as an undergraduate student, she's putting out competitive stuff. She beat out a graduate student for that research award to go there. So there's some high quality stuff you can do as an undergrad, and she works on average 6 to 10 hours in that lab a week. So it's been a continuous focus for the last year and a half for her. And she picked up a number of prizes, and I bought her a ginormous ice cream. So she's happy. This is the K-State today. So Maya joined my lab last fall, and she's doing well as well. She's working on the Ebola vaccine that was used in West Africa to treat patients recently, the one they rushed to clinical trials and actually used effectively with humans, had not been tested for agricultural spillover. And what that means is it's actually a vaccine that's built on a virus. So it's another virus that can infect humans but doesn't give them any disease. It delivers the antigen or the protein for Ebola. Your immune system responds, and then you're protected against Ebola. So it's a marvelous thing, right? But the particular virus that it's vectored on is also a disease in horses, cattle, and pigs. And although the particular virus that the Ebola vaccine is built on is considered to be attenuated, meaning of lesser disease import, we still need to check, double check, whether it can affect horses, pigs, or cattle. Because think of this scenario: if we're going to vaccinate a human population, and that human population has pet horses, or it's a farmer with cow, goes out and works with their animals, and if that human is shedding a virus that doesn't hurt the human but could potentially hurt the animals, it's a problem. And in this case, it's not so much that this is a deadly disease even in horses or cattle, but the problem is that it looks like another disease, which is a foreign animal disease in this country. It's called foot and mouth disease. And foot and mouth disease will shut down our economy in terms of our agriculture. If our cattle get foot and mouth disease, the same sort of trade barriers I talked about earlier for prion diseases in England would arise in the United States. We wouldn't be able to export our beef. For Kansas, that would be catastrophic. So this particular disease, when you see it clinically, looks like this much more important disease. So we just don't want to have a chance of spillover at all. So we're under a contract right now at K-State to do the research where we're checking to make sure that this particular vaccine virus that could be given to humans in the event that there's any sort of risk of Ebola won't actually spill over into any of our agricultural animals of interest that are affected by it. And so Maya's been involved with me, and she's already done a poster. Here's her poster. She presented this on Sunday. Again, she's looking for the virus in the tissues, and it's a little hard to see from here, but there's some blue blobs and some red blobs here. This is identifying the virus. A little clearer here where the little white things are here. But I just wanted to show you the poster at a sort of glance to show you the level of work that she's doing. This is a freshman at K-State, so this isn't too much further along than you folks. And she's in the Developing Scholars Program, which is a program for underrepresented students and also minorities. So if you come from a family where no one's ever gone to university before, or if you come from an ethnic or other classified minority, there's a whole program at K-State to encourage people to stay and to focus on research. There's lots of options as well. And then the final student I have in my lab is Katie, and she's working in conjunction with me on the pneumocystis, so fungal pathogen in the lung. And she's been identifying it. This is actually her work. This red are basically pneumocystis cysts inside the infected mice. And we're starting to look at, so where are these organisms going in the lung? What cells do they like to hang out with? What damage might they be doing to those cells? And so that's her whole interest. And the reason this is important is because these patients who get pneumocystis pneumonia, we have treatments for them, but the treatments fail. So we put them on antifungals, and then after so many weeks, the treatments fail, and we don't understand why they fail. We don't understand this organism well enough. This is an organism that they just did the genome sequencing for this past year. The paper came out. And so I worked with the gentleman who was the anchor on that paper, and that sequencing of the genome gave us all sorts of insights into proteins that we just don't know what these proteins do. So we're starting to look at these proteins and understand what the organism may be using them for. We definitely think it's using some of them to cloak itself and hide from the immune system and from the treatments. And so we try to get to the bottom of that. So Katie's working on that with me, and she's learned a number of techniques, and she's working on a project now where she's going to use this marker in conjunction with another one to see what's going on. And finally, Monica is my technician. So she's across all of the undergraduate students. She's aware of all of their projects and she works with them. But she also does some of her own work. So these are cells that are infected with RIF that she's lit up quite nicely over here. The blue is just the nuclear marker, so that's the nucleus of the cell. And then over here, we start using multiple colors. RIF is purple, and brown is a marker for a certain type of leukocyte. So what that picture shows is that not only is RIF present, but it's particularly inside this cell here. And so then we can look and see. So those types of things that undergraduates do in my lab, and then they can take these skills where they want to go next in terms of, Katie, for example, has a career goal to go off and do a certified pathologist assistant career, which is basically a master's level program. Whereas Maya's undeclared and Lizzie is going to be in the class 2020 for DVM. So some of them go off to become DVMs and some of them can take other careers. And so these are my kids. Start early. I know you're not this young, but start early with science fairs. Take advantage of all the opportunities you have. This was then in kindergarten. This was then this year, first grade. This is the Wamego Regional Science Fair. We don't go any further than that, but I know there's lots of opportunities beyond for you. And I threw this slide in just this morning, and what I wanted to emphasize is that pathology is a wide field. You can get into pathology through many paths. In most cases, you need an undergraduate degree. But then there's a master's approach. You can become a certified technician, an assistant. There's different levels where you just need masters to get there. So you do specialty training at that level. You can go and get the professional medical degrees, MD or PhD or DDM. I suppose there's dental pathologists out there too. I'm just not familiar with the field. You can also go the traditional PhD route, and that could be in biochemistry, biology, or any other related field, and then focus on your PhD topic for the pathology question. So lots of different combinations, you can get there in lots of different ways. And if you're interested, these societies here, there's 3 of them, they've got very simple URLs, acvp.org, acip.org, and histochemicalsociety.org. They all support trainees. They have information on their website about what's this career. The first one is the Veterinary Pathology Organization. The second one draws across veterinary pathology, medical pathology, and PhD type pathology. And this last one here is much more focused on the techniques. So if you like the idea of a kitchen at the bench and doing all sorts of things with chemistry and making things appear and identifying things in tissues and cells, that's a great place to go. And they have, they actually paid for Lizzie to go to San Diego, that group down at the bottom. And one other thing I'd like to show you is that in Chicago, experimental biology is one of the major conferences that's every year in the United States. It goes across anatomy, physiology, biochemistry, and pathology, a number of different subjects. But in the ASIP section, which is Investigative Pathology, there's a special program for high school teachers where also students go with their teachers. And they pull mainly from the location where the conference is going to be, but more and more they're starting to offer scholarship options to bring in people from other states. And so this program is on the, it'll be on the ASIP website, as well as this is from the meeting in Boston a couple of years ago. But it's called Sleuthing Science, and every year they pick a really neat human pathology topic and they delve into it and they do a lot of hands-on workshop type stuff. And then you get a pass to go to the exhibit floor and explore the conference. So it's a great opportunity. And that's everything for me. I'm a little over, which I apologize for. But if you guys have any questions, I'll hang out afterwards too. So hopefully it was entertaining and informative.